really discovered the DNA molecule. Did you know that this molecule was discovered in 1969? It is not that long ago. Okay, perhaps a bit longer for you than for me. Now, we want to see that special code on the DNA that cause the code how to make a protein. And we call that specific part that is telling the body how to make a protein a gene. Yes, we are going to see how the genetics and heredity happens with the DNA molecule. So when we look at the history, it is important to know that genetics is the study of genes, and inheritance or heredity is how traits or characteristics are passed on from generation to generation. How do we inherit my mother's blue eyes? Or how can I make sure, if I'm a farmer, that the good traits or characteristics or quality of my show bull is passed on to the calves that he sired? Perhaps I am a vegetable farmer, or if I'm harvesting oranges each year, and I want the offspring to be just as tasty as the tree that I am giving water and pampering all year. How does that happen? Chromosomes are made of genes, and genes are built up of DNA. So when I am looking at the genetic material, I'm looking at genes and chromosomes and DNA, and we find that inside the nucleus of a cell. Gregor Mendel is considered as the father of genetics, and we are going to look at the reason why. So when I look into the nucleus of every cell, I will find the chromosomes. If you have to show me a picture of chromosomes, it will be a chromosome that has been replicated. So chromosomes sort of look like this. Now, it contains the genes, short parts of the DNA, and it is made up of DNA, thread-like parts that contains the genetic code for a specific protein is a gene. It's made of DNA. So, here I have 23 pairs of chromosomes. Pair number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23 chromosomes. 23 pairs of chromosome. This is called a karyotype. A karyotype is a photographic arrangement of a complete set of chromosomes in a human cell. So, we already did this, a bit of revision. A homologous chromosome means a chromosome from the father, a chromosome from the mother. And I would like to use symbols so that we do remember. Let's make the green one a chromosome that came from the dad, and the purple one that was part of the mother. when replication takes place, I have still a homologous pair of chromosomes. This is still coming from the father, but it replicated. And this came from the mother. Now I call this part a sister chromosome, and of course the centromere will keep the chromosomes, the two parts, the chromatids, together. So when I am looking at a homologous chromosome, one of a matching pair of chromosomes, one inherited from each parent. So this is a chromosome. And if I look at this picture, I have a homologous pair, but I can see this was not yet replicated. But that is not important now. What is important now is on this specific spot, which is called the locus, I find the color of the flowers gene. This is a gene for the color of the flower. On the homologous partner, I find the gene for the color of flowers. So this is telling me it's a plant chromosome. You agree? Now, on the one, it says purple flowers, and on the other one it says white flowers. So we are talking of the alleles for flower color. The one allele states it is purple flowers. Other allele on the same locus will tell me what is this chromosome's gene for flower color. Please go and quickly revise what does the word allele mean so that you can define it. Right. So now when I have a homologous pair and this homologous pair had replicated, I can see they are a pair, they are exactly the same size, and they will have, if this is the alleles for flower color, this will also be the alleles for flower color. It may not necessarily be the same. It can be purple and it can be white flowers. If we are looking at a human homologous pair and we say that this is the alleles for eye color, this will in the homologous pair be the one for eye color. They can both be brown eyes or they can both be blue eyes. Or this one maybe can be brown eyes and this one blue eyes. So, what is the color of this person's eyes? Is it brown? Is it blue? Let's see if we can discover something else. So, alleles, alternative versions of the same gene. The gene for a particular inherited characteristics resides at a specific locus or position on the homologous chromosome. So, if I look at this picture, this will be called alleles. Why? They are for the same trait on the same location on two different chromosomes. But if I have a spot there and a spot there, it will tell me that it is two different genes. It will code for two different characteristics or traits. Now, a genotype and a phenotype. We have to now see what is the difference between genotype and phenotype. Genotype is telling me what is on the genes. So, on the genes, I will have allow for green eyes. So, I think that can be eye color. But I have eye color here on the genes. That tells me the genotype of this organism. On this, I will have also a specific gene for body shape. And I will have a gene that is telling me that this organism has a specific number of legs. So, this is telling me on the genes what is this animal traits. But the phenotype is what can I physically recognize about this organism. I can say it has green eyes. You agree? It has a blue body. It has 3 legs and it has wings. Okay, and this was all codes that was on its genes. So when I'm asking the genotype of an animal, I use codes. When I ask the physical phenotype, I say it has green eyes. But when I want the genotype, I will have to discover a bit more. So, if I take the gene for purple flowers, you remember we had a gene for purple flowers. Now we use a capital P to indicate the gene for purple flowers. Why capital? Because purple is the dominant trait. And then small p will mean what? It will mean white flowers. But I still use the small letter of the dominant trait. And white will then be the recessive gene. Okay, I really hope that you are still with me. So, I have here a dominant feature, therefore I've written it in a capital letter. I have a recessive feature, therefore I've written it in a small letter. But I use the P because the dominant feature is purple. In this case, we say the recessive feature is white. And when I have a capital P, small p, gene combination, we say it is a heterozygous, different genes. This specific organism has 2 different genes for, let's use flower color. Okay, but what happens with these alleles? Locus for second gene containing alleles has a capital Y and a capital Y. Yes? So, we say it is homozygous in its genotype. Let's say that capital Y was for round seeds. Round seeds. And round seeds is dominant. Then it means the genotype on both the alleles are capital Y, so it has the same genes on both the loci, and therefore we say the genotype for seed shape is both dominant is round seeds, but it is heterozygous for flower color. It do have a purple dominant gene and a white recessive gene. Therefore, two different genes is a heterozygous genotype and a homozygous genotype. So, if we go back to our little explanation here, is the genotype will be a code, right? And the phenotype will be what we physically see. And now I wonder if this is the genes for this flower to have offspring. There cannot be only a male, but there's also a female. No, but paternal, that is the dad's chromosome, and this is maternal, pa and ma, paternal and maternal, and these two are going to have babies. So, what will happen to the flower color of the babies if this is the mother and this is the father, and what will happen to the seeds? Okay, so definitions. Genotype, the types of genes or alleles that is present. What is the gene of the father? Purple. Dominant purple. What is the gene of the mother? White. Now, they want to have a baby, and these two genes will come together. Where? After fertilization took place, yes, the ovum and the sperm fuse and forms a diploid zygote. And now, the new offspring has a dominant purple gene and a recessive white gene. The question will always tells you which one is dominant, which one is recessive, or will give you enough information to deduce which one is dominant and which one is recessive. It's important to find that out. Now I know the genotype of our previous slides offspring. But the phenotype is what will it physically, what will it physically look like when we look at that offspring. If a characteristic has same alleles, it is a homozygous gene or characteristic. If it is heterozygous, I know that it has a purple and a white gene. Right, heterozygous characteristic. Okay, so let's look at dominant and recessive again. Dominant means that this allele will be expressed if it is present. If a dominant allele is present in the genotype of the chromosomes, it will be expressed. And we always represent it with a capital letter. The trait we are looking at is freckles. Let's choose freckles for our letter to represent the dominant allele and the recessive allele. If you have freckles, it is a dominant trait. If no freckles is on the gene, it will be a small f. So recessive alleles will only be expressed in the absence of a dominant trait, and we represent it by a lowercase letter. Try to repeat. How do I define dominant? The allele will be expressed always when it is present, and I look for it or write it as the capital one. A recessive allele will only be expressed in the absence of the dominant one. Always expressed if it's present, only expressed if there is no dominant trait. Okay. So, a dominant allele is expressed even if it is paired with a recessive allele. Here, I have a phenotype. Phenotype. This cat is brown. Yes. This cat is brown. Yes. And this cat is gray. Okay. So, now I'm looking at the genotype. This cat is homozygous, capital B, capital B. You agree? Its genotype is homozygous brown. This one is homozygous gray. Can this cat shows any other color than gray? No, because the recessive alleles is visible. Gray is recessive over brown. Okay, because I see small letters. If this cat is gray, it can only be homozygous for the recessive trait. But this one is, the phenotype is brown. But can I say it is capital B, or capital B, small b? I cannot say unless I have more information. Now, so this one is a heterozygous genotype. But physically, I will see only the dominant allele because it is always expressed, even if it is paired with the recessive allele. If I see the recessive traits, it means it is small b, small b. So, what did Gregor Mendel then do? He was an Austrian monk, and he lived in a monastery, and he started to experiment with pea plants because he recognized that there are specific characteristics that occurs in some plants and then in others it is gone. Why pea plants? They were available, they easily grew in the garden, they reproduce quickly, and they have differences in their traits. So, understood that there was something that carries traits from one generation to the next, and they call it a factor. During Gregor Mendel's experiments, he was not aware of the existence of genes and DNA and chromosomes. He only knows there was a factor that was available. There you see Gregor Mendel, 1800s. Okay. So, he started his experiments to apply the question on inheritance. For 7 years, Mendel bred pea plants and recorded inheritance patterns in the offspring. Particulate hypothesis of inheritance. Parents pass on to their offspring separate and distinct factors. Today we call it genes. Parents pass on to their offsprings separate and distinct factors that are responsible for inherited traits. Now, Mendel was very, very careful to make sure that there are no contamination. It is very difficult to know which plant is the father and which plant is the mother. Which flower had produced the pollen, because the pollen contains, not is, the pollen contains the male gamete. And inside the ovary of the flower is an ovule with the female gamete. So what he did was, he worked very carefully. He tried to enclose the plants that he was working with. He removed the stamens, in this case, from the purple flowers, so that this purple stamen will not produce pollen and self-fertilize this flower. He took that and he transferred it with equipment, delicate equipment, to this the pollen from the white, and he put it on the purple ones. He knew exactly where did he do this pollination and made sure that the carpel matured, and then he planted that, because when you have this, you don't know how the offspring will look. No, this is only containing the embryo of the new offspring. So you have to plant it, take care of it, and now you have your first generation of offspring. So purple and white. White was the father and purple was the mother. And the first generation offspring then was there. He used color of the flower, position of the flower, seed color, seed shape, pod color, stem length, all different features that he used. And it was very important to prevent self-fertilization. Okay, so he cross-fertilized his plants by hand. It was very important because he needed to control who was the parents in order to see if the parents transfer the genetic characteristics. Now we come to what will happen. The first thing is we are looking at monohybrid crossings. That means we are only using one characteristic at a time, not purple flowers and what pot shapes, and we only look at one characteristic that we are crossing. Now, what happened here was the purple flower with the one flower. The parental generation was true breeding, but we rather use the word homozygous. Homozygous, and we will use the capital P for the genotype, with the small p, small p. And all the plants have purple flowers. All the plants have purple flowers. And then he took all these purple ones, which then had 3/4 purple in the second generation. So this is the self-fertilized. They fertilize themselves with their own pollen. And the F2 generation showed 3/4 of purple flowers and 1/4 of white flowers. Now, what happened was he counted them and he had purple flowers, white flowers. All the first generation was purple. But the second generation had 705 purple and 224. That roughly gives us a ratio of 3:1. Now, if we look at humans, we can see there are quite a few resembling. These are brothers. I think the one was the father and brothers, but we can see that they really look alike. Here we have a father, a son, and his son, and we can see how they look alike. This is father, next generation, and last generation, and we can see the traits. And here I have quite a familiar picture to me, and so we are not twins, no? And this is my son, and we can see there are definitely features that belongs to all three of us. So, what happens during this law of dominance? In the monohybrid crossing, mating of two organisms that differ in only one characteristic, that is monohybrid, the recessive disappear in our first generation. Did you see that? Homozygous purple, homozygous white. First generation, all of them disappear. But in the next generation, we have a 3:1 ratio of dominant trait to recessive trait. Right. And I would like to explain that to you now on a diagram, and then I am going to leave you so that you can answer some of these questions yourself. So, I am going to write in yellow, and I want you to do the next calculation. I have a purple, and it crossed with white. Okay? If this crossing takes place, it means all the offspring will have what genotype? All the offspring will have a genotype of capital P, small p. You agree? Okay, so how do I calculate it? Last thing, I promise, is I use a Punnett square. I use a Punnett square by making a diagram showing the probabilities of the outcome. So I am going to draw a square, and here I'm going to write down the father. The father was small p, small p. I separate this because I know on a gamete there will not be two but only one of the genes. And because it's in a gamete, I make a circle. But the other gamete will also have a small p. Due to meiosis, the two alleles of the father will be separated to form only one of these alleles will be possible on his sperm. In the female, and she was capital P, there will also be meiosis taking place, which will form the ovum, and now I will have a capital P. And because it is gametes, I have to circle them. Now, if this gamete and this gamete fertilize, that is the possible offspring. There, and there. The only probability that can happen with this homozygous white flower and homozygous purple flower is a heterozygous flower. But what is the color of this flower? Definitely purple. You agree? Definitely purple. I hope you follow me. But now, what will happen if I have in the second generation this dad and this mother? I draw my Punnett square, and I knew due to meiosis, it can either be that or that for the father gamete. On the mother's gamete, it can be this or this. So, if these gamete and this gamete forms the offspring, capital P, but if this gamete and this one forms the offspring, this will be the alleles of the offspring, or this one and this one, or this one and this one. Please remember, you have to put circles around that. You have to say, this happened by meiosis to form gametes. Right. And now, let's show which are purple. That one is purple. That one is purple. Of course it's purple because the dominant allele is present. And this one is purple. So I have 1, 2, 3 purple flowers. 2 white flowers. I have 1 white flower. And now I'm not still on the board. So my ratio purple to white in my second generation, my F2 generation will be 3 to 1. And with this, I'm going to stop our lesson. Read through your textbook. Try the questions that I have added. Remember to concentrate, and we will continue this very interesting one with an example of a brown-eyed dad and a blue-eyed mum. What will be the color of the offspring? Thank you very much. What do you think we have there? We have bacteria, and we are going to look at how bacteria adapted themselves due to selection pressure. Pressure that came from their environment and how they changed to survive. We look at antibiotics. Yes, you all know that antibiotics was developed by Alexander Fleming in the 1928. He discovered it. What happened was he had a Petri dish full of bacteria, and accidentally there was a piece of fungus, penicillin fungus, in between the bacteria. He then realized that the bacteria stopped growing, reproducing, and they thought it was the presence of the fungus. They explored it, and so they made antibiotics. During World War II, many soldiers were saved from huge infections that was due to wounds that was from the war, and they start to treat bacterial infections with antibiotics. Tuberculosis, gonorrhea, syphilis are some of the bacterial infections that we have studied throughout our grade 11, grade 12 biology career. Antibiotics are successful in controlling bacteria because the one that we know, antibiotic penicillin, can make the cell wall of the bacteria grows weaker. When the cell wall is weaker, it cannot withstand the turgor pressure inside, and the cell wall bursts due to the pressure inside. That is why bacteria are being killed by antibiotics. Why not killing a virus? Oh yes, we know. A virus do not have a cell wall. A virus has only a protein coat. And therefore, the antibiotics that can kill bacteria cannot destroy a virus. Antibiotic resistance. We can explain it like this. Bacteria are able to survive and grow in the presence of one or more antibiotics, and they can continue to cause the infection. If that happens, it is telling us that the bacteria got resistant. If they continue to survive and grow in the presence of an antibiotic, then they are resistant. We must remember other microbes like viruses and fungi can also become resistant to other antimicrobial drugs. A microbe is a microorganism, and there are various drugs made to kill viruses, fungi, and bacteria. Antibiotics only for bacteria. Remember that. Without effective antibiotics, common infections such as bacterial pneumonia, then you cannot breathe. There is a bacterial infection in the covering of your lungs, and it is life-threatening if you cannot breathe and take up oxygen. Now, other things like operations. After any operation, you will get a series of antibiotics to prevent any bacterial infections due to the fact that during an operation, your immune system is under pressure. And all these operations, open heart surgery, will be very dangerous, and deaths from infections will be more common. If the antibiotic cannot kill the bacteria, if the bacteria are able to survive and grow in the presence of the antibiotic. I want us now to look at a video that explains this bacteria versus antibiotics.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere, on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that helped them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria like Salmonella even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria, and although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections can have a major impact by keeping more non-resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de-escalation may sometimes work better than an evolutionary arms race.
So if we need to explain how antibiotics become resistant, we can say that we have a lot of bacteria in our body that cause an infection. Antibiotics will kill the bacteria that cause the infection, but if there are drug-resistant bacteria that can grow, especially when we do not complete our course because we think I am well enough, or we share the course of antibiotics with someone, then there are some of the bacteria that stays alive, and they are normally the ones that develop the resistance. And we also see that it is not only those that develop, but they can also pass their resistance on to other bacteria. And now you drink the bacteria, but they stay alive. And we know that bacteria are very, very fast reproducers. Now, some plasmids enable the bacteria to produce an enzyme that can make antibiotics useless. When the plasmid is inserted into other bacteria, like we saw, they can pass it over. It can spread easily, the antibiotic resistance, and quickly among bacteria. Genetic material spontaneously changes. We see the mutations cause the antibiotic resistance. Now, it is not some miracle. It is because of a mutation. When can mutations occur? When not all of them die. So we need to make sure that all of the bacteria is dying with the intake of antibiotics. Okay, so resistant and non-resistant bacteria exist. Bacteria multiplies. Some of them are resistant because they did not die. They can still keep on growing, and they transfer even their gene. And now all the others that are being killed. How will they be killed? Their cell wall becomes weaker and they burst. Okay, but if there are some that can multiply and thrive, not ever again will the antibiotics, this type of antibiotics, work in this person, because this bacteria now are resistant. Because of the misuse, not completing your course, people stop using when they feel better, or they use antibiotics, but they do not even have a huge bacterial infection. The mutation takes place, and the course do not kill all the bacteria. So some have a mutation, and those that are resistant stay alive. Natural selection caused them to survive because their environment changed. They are adapted, even through a mutation, they adapt to the environment. And the antibiotics provide selective pressure. Selective pressure is the factor that causes this bacteria to survive. So the antibiotics was the selective pressure. So the survival pass allows of resistance to the offspring. Very important. The frequency of the allows need to increase so that there are enough survivors. That is a very, very important sentence to put into this answer. The frequency of the allows need to increase at such a rate and in such a quantity that there are enough survivors, so that the successive generations can increase in number. And now, if The antibiotics are prescribed for the bacterial infection, and it is given to a person with all this resistant bacteria. The antibiotics will do nothing to kill the bacteria. The bacteria will still grow and survive and keep on with whatever this infection is doing to the body. That is very sad.
Hi beautiful souls, this is Nikita Winkler and I am here to share easy tools for stretching that you can do just about anywhere. In this session, we will be doing mobility stretches. This session is great for you if you want to wake your body up or warm your body up. Please remember that this is a demonstration video. So what's most important is that you take your time. So let's get started. So what we're going to do is we're going to create circular movements in your wrists, your shoulders, your torso, your hips, and your knees. We're going to go one way and the other. So let's get started. Interlace your fingers and rotate. And change. And then we go into the shoulders, big swings in the arms. And change. Remember to take your time. And you can also place your hands on the shoulders, create your rotations. Or if you don't like any of those, just rotate the shoulders back. From there, chest goes front, side, back, side. Front, side, back, side. And now we go for a full circle and change. Into the hips, one way. You can start small and keep increasing the range of motion. Last, our knees. Bring the feet together and hands on the knees and rotate. All right, don't forget to go the other side as well. Thank you for joining me. Don't forget to breathe. Stay present and happy stretching.
Win free airtime with One Africa TV. Share your comments on the channel. SMS your feedback, name and town to 555. One Africa TV. It just gets better.